Microtubule-associated protein tau, its biological role and pathological form. Hi, my name is Irving Vega. I'm an associate professor in the Department of Translational Neuroscience at the College of Human Medicine at Michigan State University. In terms of disclosure, I have no financial interest or compensation to declare. At the end of today's lesson, the expectation is that you're going to be able to distinguish between different tau isoforms, recognize the functional domain of tau proteins, distinguish known tau biological functions, identify relevant tau postulational modifications, and understand the regulation of tau proteins. Differentiate pathological tau structures, and at the end, get a comprehension of the transmissibility of pathological tau species. It's important to emphasize that this is an overview, and you might need to take further reading to understand the nuances and details of the brief introduction that I'm going to be making today. In terms of the gene, the microtubule-associated protein tau gene, which is the nomenclature used when you're referring to the gene MAP. There are two haplotypes that are observed in the population, namely H1 and H2. And the production of these haplotypes is by inversion of 900 kilobases in the region of chromosome 17 that includes the MAP gene. In this illustration, the H1, you can see the location of the MAP gene. There is an intragene called cytoin that expresses a small protein that we don't going to be discussing today, just to mention that there is that intragene within the MAP gene. And then the H2 is the inversion of that 900 kilobases, where the location of the gene and those other genes within those 900 kilobases are inverted. H1 is the most common among whites. There are different subhaplotypes that have been identified. The one of high interest is the subhaplotype H1C that has been associated with tauopathies, which are collectively neurodegenerative diseases that have pathological tau, namely progressive supranuclear palsy, corticobasal degeneration, and Alzheimer's disease. The H2 haplotype is rare in America, mostly found in Europe, and differs from H1 in addition to the inversion to a deletion of 200 base pairs in intron 9. That is very illustrated here, where you see the H1 with a deletion of the 200 base pairs within the intron 9, and the H1 that has those 238 base pairs. In addition to the deletion and the inversion, there are specific SNPs of single nucleotide polymorphism associated with H2 and H1 that has been identified. The relevance of these haplotypes and the SNPs are still to disease and biological function are still under study or poorly understood. From the gene, from the MAP gene, again in chromosome 17, that encompasses 16 exons. Through the process of selective splicing, there's a specific exons that are selectively spliced out and not represented in the mRNA, namely the 4A, 6, and 8. But there are also specific exons like 2 and exon 3 and exon 10 that are also selective spliced and generates different isoforms that we're going to talk in a second. The gene expression of MAP is found in the central nervous system, the peripheral nervous system, and all involve neurons and glia cells. In the peripheral tissue, the expression of the MAP gene is found in retina, skeletal muscle, testes, breast, kidney, heart, and adipose and soft tissue. Recently, there has been interest in the expression of one isoform that I'm gonna discuss in a second in the heart due to the generation of cardiomyopathy associated with the expression of that tau isoform. From that mRNA that leads again to the production of different tau protein isoforms generated by selective splicing. The main isoforms discussed are these 6 isoforms present in this illustration. And those 6 isoforms are generated by the selective splicing of exon 2, exon 3, and exon 10. Exon 10 is within the repeats, the 4 repeats present in the microtubule binding domain, and the presence of the exon 2 generates the repeat 2 that leads to the isoforms that have 4 repeats. That's what the 4R comes into play in the naming of the protein tau. The presence of exon 2 and 3 leads to the protein tau N2N4R. That's the splicing name, but then the clone is tau 40. It's important to mention here that in the literature you will find different nomenclature to refer to these tau isoforms, but namely it will be tau 40 or 2N4R or 3R. Important to mention here is that you're never going to see the exon 3 alone or included in the absence of exon 2. So that generates 3 different isoforms, either with 2 and 3 exon including exon 2 and 3, with only 2, or without exon 2 and 3. That is what is referred to the 2N, 1N, or 0N. Then the splice out, selective splice out of exon 10 leads to the other 3 isoforms that are the absence of that second repeat and generates a microtubule binding domain that has only 3 repeats, and that's the 3R that you can see here. So that in total generates the most common study and referred in the literature as the 6 isoform of tau. However, inclusion of the exon 4A leads to the production of another isoform that is expressed in both CNS and peripheral nervous system, in the central nervous system and the peripheral nervous system. Although it was primarily studied and found in the peripheral nervous system, we know now that it's also detected in the central nervous system. And it's referred to as big tau. Big tau is about 90 to 110 kilodalton, where the 6 isoforms, or referred to low molecular weight tau isoforms, range from 45 to 60 kilodalton. Even though the 6 isoforms are mostly expressed in the central nervous system, the opposite is understood about the big tau, where mostly is in the peripheral nervous system, but again is also found recently in the central nervous system. The function is still under study for big tau, but is apparently associated with as well with microtubules. But further studies are needed to really understand the differences between the biological function of big tau and the 6 isoforms. The 6 isoforms in terms of disease are related to tauopathies, where it's still unclear what is the role of the big tau, although recent data indicates that it aggregates, especially in the heart, and creating cardiomyopathy. And the association between cardiomyopathy and Alzheimer's disease is under study. Most recently, another isoform of tau has been identified, and that isoform is known as W-tau. And the isoform is formed by the insertion of intron 12 during the generation of the mRNA. And this intron 10 contains a stop codon and also a polyadenine sequence that generates a functional mRNA that leads to the production of 2 proteins. One that is known as W-tau or WT30 or WT42. The W is added because the sequence that is added contains tryptophan, and that W refers to that. This work is being done, or the first group to report this isoform is the group of Jesus Avila in Spain. The W30 is actually the isoform that contains that intronic sequence inserted in the mRNA that leads to a protein that has a splicing of the exon 2 and 3 and exon 10. So it generates a protein that is 3R0N with the extra sequence that comes from intron 12. But there is also detected a protein that they name WT42, which actually contain the two exons, exon 2 and 3 in the N-terminus, and the 4 repeats in the microtubule binding domain, in addition to that intronic sequence that is added that has that stop codon at the end. Interestingly, in vitro data shows that sequence added to the tau promotes or is sufficient to inhibit the aggregation of tau proteins. And these WT30 and the WT40, contrary to the 6 isoform, these isoforms do not aggregate in vitro. In addition, recent studies show that the peptide itself can inhibit the aggregation of 6 of the other tau isoforms in vitro, but also not only tau, but also the A beta peptide aggregation. Again, this is work done by Jesus Avila group in Spain. They also correlate the expression of the abundance of this protein in Alzheimer's disease, where the abundance of this WT30 and T42 is reduced in Alzheimer's disease. And the researchers speculate that the reduction of this protein is actually associated perhaps with the aggregation of the other 6 isoforms. Interestingly, they show that the WT isoform, the VW tau, is mainly expressed in regions that are resistant to pathology in Alzheimer's disease, such as the cerebellum. However, it's important to emphasize that this is new study, recent studies that need to be further, this isoform need to further study about their role either in the pathology of or their neuroprotective role. The main domains on the tau proteins are the N-terminal projection that includes the acidic region in the tau protein. And this N-terminal projection is being associated with mediating signaling pathways for tau protein. And the microtubule binding domain will contain the proline-rich domain, which actually has been studied as a region that has an effect on tau aggregation. And then the repeat domain, which is the main area of the protein that promote the interaction with the microtubules. And I should mention now that is the minimal region that is required for tau to form aggregates. So this region is actually the main region that promotes tau aggregation. So the main biological function, or I should say, the function that was best studied and best known for tau is regulation of the dynamics of microtubules. And it has the repeat domains is the one that sits and interface with the A and beta, alpha and beta tubulin heterodimers. And then the projection domain extends out from that interaction of the microtubules, and the speculation is that serves to create a spacing between the microtubules. Other proteins also known to associated with the microtubule binding domain or repeats are F-actin, HDAC6, and apolipoprotein E. And the microtubule binding repeats associated with binding to lipid membranes and also to bind both DNA and RNA. In terms of the proline-rich domain, is actually known to promote protein-protein interaction, and is one of the regions that also, in addition to the acidic region, that has been speculated to promote or to be involved in these other functions other than microtubule dynamic regulation or dynamics, to be involved in signaling pathway, like interacting with FIN, PI3 kinase, and again associated with regulation of neuronal cell signaling, binding to DNA and RNA in terms of regulating the nuclear function. Tau proteins are found both in the cytosol but also in the nuclei. The role in the nuclei is still poorly understood. It actually mediates also binding to actin in addition to maintenance of the microtubule assembly. In terms of the acidic region, involved in signaling cascade, regulation of the protein phosphatase 1 or PP1. It also regulates the subcellular localization of the tau protein and facilitate the interaction with the plasma membrane such as annexin A2. Even though tau, I should mention that in addition to understanding that the main biological function was microtubule dynamics for tau, now we also understand that tau is not only localized to the axon, they're also localized to the dendrites, to the nuclei, and also in the soma. And the role of tau in these other places, especially in the dendrites, are associated with synaptic transmission. Tau actually, the acidic region, promote protein-protein interaction with apolipoprotein A1 in the absence of the two exons 2 and 3, beta-synuclein and synaptophysin. So the domains not only dictate their biological function, but also their role in their biological role. In terms of regulation of the protein, tau is heavily post-translational modified. So there are many different post-translational modifications identified on the tau protein, mainly phosphorylation, which is associated to both biological function and pathological function. Tau is a phosphoprotein, but the aberrant phosphorylation of tau is what leads, we understand leads to a transformation in the conformation that leads to the pathological forms. But there is also ubiquitinylation, glycosylation, sumoylation, acetylation, methylation, lipidation, hydroxylation, and even disulfide bond formation. So tau is heavily post-translational modified, and how the combination or the timing, a temporal formation of this accumulation of this post-translational modification and the impact of those modifications in terms of pathological transition from biological to pathological role are currently heavily studied. In addition to a specific addition of molecules to particular post-translational modification to tau protein, there is also cleavage of the tau protein by specific caspases, calpain, for example, that can actually generate truncated forms of tau proteins that leads to the formation of toxic fragments or aggregated forms of tau. There is some truncations that are known to take place before tau is aggregated, and another truncations that take place after tau is aggregated. So you should refer to the literature to find the dynamic of those known proteins that especially caspase 3 and caspase 6 and caspase 3 that leads to aggregation of the protein, or the product of those cleavage are associated with pathological forms of tau. So in terms of protein structure, is understood that accepted that generally accepted that tau form is sort of a paperclip conformation when it's free in the cytosol, and that actually changes when it bounds to microtubules, where the repeat sits on the microtubules and the projection domain and then stand out. Within the N-terminus, there is that PAD region of phosphatase activation domain regulation that we mentioned before for PP PP1, and how that is exposed during disease or biological role is under study. So the transition from these biological structures to pathological forms is something that is, and what is under study, and what is the role of post-translational modification on the transition of this paperclip to this more open structure, and how the tau proteins come together is something that is heavily studied in the tauopathy field. So in terms of pathological forms, when you look at a neuropathological analysis, you find neuropils or neuropil threads that you find the tau aggregated. You have the formation of what is known as neuritic plaque, and then you have that frame shape of neurons in the somatodendric compartment that is neurofibrillary tangles. And those are the pathological signatures that you see when you look at postmortem tissue of in particular in Alzheimer's disease. Most recently, those structures have been actually identified in have been studied by cryo-EM, and these are recent studies of purified aggregated forms from tissue, from AD, Alzheimer's disease, from CTE or chronic traumatic encephalopathy, peak disease of frontotemporal dementia, and corticobasal degeneration in this case. And cryo-EM has given some idea of how the conformation of these aggregates in different diseases, to a point that now is referred to different tau strains, and the conformation of tau differs between the different diseases. However, there is a limitations in cryo-EM studies. But looking through the axis of the aggregate, there is the C shape that is characteristic of that interaction of the repeats. Remember, I mentioned that the repeats are the minimal region that is required for the aggregation, and is the one known to then promote the aggregation of the tau protein. So that C shape is mostly characterized or found in AD and CTE, although there's divided then in two types, type 1 and type 2, based on the orientation of the C shape and the interaction between repeat 3 and repeat 4. Then in peak disease, you have the repeat 2, and then you have the R prime, and it's more open conformation of those filaments, those ultrastructures purify from postmortem tissue. And then in CVD has a more open structures than the C shape that is seen in AD and CTE. Again, the limitation of cryo-EM is that not only is an average of the structure, it don't give specific details about amino acid level. And also there was reported that there was some unexplained dense region within these structures that still are unresolved. There were only few brains that were used to extract these aggregates, and these aggregates during this extraction of insoluble, or what is called insoluble tau, may only represent a fraction of what actually is found or is pathologically related. Another piece of information that is crucial is that these differ. There is a difference between the structure detected in cryo-EM or resolved in cryo-EM from extracted tau from tissue versus the tau that is actually induced by recombinant tau protein using heparin. So that is what is studied in vitro versus what is found in tissue might be relevant to further study whether in vitro tau aggregation is relevant to disease or not. So in the process of tau aggregation, as I mentioned before, it goes from monomer to dimers, and then from those dimers to trimers, and those are the combination of those molecules, which I still don't know how many tau molecules are in a tau oligomer. those oligomers then come together to form filaments. And then those filaments come together to form then the neurofibrillary tangles. So right now, the toxicity, what is understood is that these oligomeriforms tend to be the most toxic ones versus the ones that go into higher structures from filaments to tangles. So these are less toxic and perhaps neuroprotective. So the rationale between that is just thermodynamics. Where is the molecules that have higher entropy versus and low entropy versus low I mean high entropy and low enthalpy versus high enthalpy and low entropy. So you have those that have higher or more dynamic structures that are the oligomers, have more energy and may induce more toxic effects than those that have high enthalpy, are organized, and for that reason then they have less entropy. So it's mostly a thermodynamic explanation. Lastly, tau transmission and spreading is actually based on the progression of pathology in the brain of Alzheimer's disease and other tauopathies, where is understood that tau molecules, and these have been supported by in vitro and also animal model study, where oligomeriforms of tau are injected in the tissue and are shown to prime the further aggregation of endogenous tau. So there are different, however, that process, how tau is released from one neuron and uptake from another neuron, is still unknown. But there are several hypotheses. One, that tau is actually released from one neuron to neighboring neuron through exosomes, or they are secreted out, and there are specific receptors that can actually help in the uptake of that pathological tau. Or there is through the presynaptic synaptic activity, or even nanotubes, that are membrane nanotubes between cells that actually are hijacked by these pathological forms, and then they can actually go from one neuron to the other one. It's still unclear whether this is a normal function of the tau protein that then when there is an increase of pathological structure, use the same system or not. So further studies are required to understand this spreading or transmission of tau or pathological tau that is observed passing from one neuron to the other one. So in summary, mainly the tau isoform, the Vega isoforms, and that we talk are the ones that are for the study, and you find more information. However, there are two other isoforms, the big tau and the W tau. And tau proteins have diverse biological roles beyond microtubule binding and microtubule dynamics. The domains indicates those multiple biological roles that are still need to be further understood. How post-translational modification of the differentiate between physiological and pathological forms, or what combinations are required to go to transition from one state and the other, is still under study. The pathological conditions, the isoforms, are different, and they go into these structures, and how oligomers are more toxic than neurofibrillary tangles. There's still something that is required further studies. And the pathological tau species and how they are spread and transmitted from cell to cell is something that will, if we understood, will revolutionize how we can treat these diseases in the future. With that, there are a few references that you should actually look at if you want further details, which are going to guide you for further information and then go into more detail on the nuances of tau later on. Thank you.